Welcome back to the deep dive. Today we are strapping in for a ride that I know many of you have been on for the better part of a decade. If you work in critical care, if you've spent any time standing around an ICU nursing station at 3 in the morning debating protocols, you know exactly what I'm talking about.
Oh, yeah.
We are talking about the absolute emotional roller coaster that is vitamin C in the ICU.
It really has been a roller coaster. I don't think there is a single molecule in the entire history of critical care medicine that has generated more hope, more controversy, and frankly, more emotional whiplash than ascorbic acid.
Truly. I mean, if you've been working at the bedside since, say, 2017, you remember the early days. You remember the miracle stories.
You do. The Orange Crush era.
The Orange Crush era, exactly. We thought we had finally found the silver bullet for sepsis. You remember protocols being taped up in nursing stations overnight, and that feeling that we were finally going on the offensive against organ failure.
And then, you probably remember the crash.
Yeah.
The recent wave of large randomized trials that felt like a bucket of ice water being dumped on your head.
It was so sudden.
It was. We went from this cures sepsis to this doesn't work to this might actually harm patients in a very, very short operational window.
It was brutal. And that leaves the clinician, you know, in a very difficult spot. You're standing there during rounds, looking at a patient in septic shock, and you have this conflicting data just swirling in your head.
Right.
Do I give it? Do I not? Is it malpractice to give it? Is it malpractice not to give it?
Exactly. And just when you thought the book was closed, just when you thought we had, written the obituary for high-dose vitamin C, here comes the PETAL Network with the ASTER trial.
PETAL Network. Now, these are the heavy hitters.
They are. This isn't just another small observational study from a single center. This is the ASTER trial, published in Chest Critical Care in 2025. The title is Ascorbate for Organ Dysfunction in Critically Ill Patients with Sepsis. And I have to say, the data in here is fascinating. It's messy, it's complicated, and it is absolutely vital that we understand it because it challenges the narrative we've been building for the last 3 years.
So here is our mission for this deep dive.
Right.
We know our listeners are the folks in the ICU, the intensivists, the nurses, the pharmacists. You guys are the ones making the decisions.
At the bedside.
At the bedside. So we are splitting this analysis into 2 parts because there is simply too much to cover in one sitting.
Yes. Today, right now, is part 1. We're going to strictly look at the what and the how. We're going to unpack the data, the study design, the biomarkers, and the safety signals.
We're being forensic accountants today.
They are. We're going to be rigorous forensic accountants of this trial.
And then in part 2, we'll get into the bedside application and the clinical synthesis. But for the next hour, we are doing a forensic analysis of the ASTER ascorbate trial. We're going to walk through the clinical problem, the unique study design because it was a platform trial, which is really cool.
Here we go.
The dosing, the primary outcomes, and then the plot twist.
The plot twist is the secondary outcomes. That's where things get really interesting.
Exactly. So let's set the tone right now. We are not here to pick sides. We aren't team vitamin C and we aren't team placebo.
No.
We are team data.
That's the only team that matters. And specifically, we need to approach this with the mindset of critical appraisal for a trial that was stopped early.
Right.
Because that is the headline here. This trial was terminated before it finished enrollment. Interpreting signals from a stopped trial requires a very, very steady hand.
Okay, let's unpack this. Let's start with section 1, the clinical problem and the biologic rationale. Why are we even still talking about vitamin C? I feel like I need to ask the basic question. Is this just about preventing scurvy in septic patients?
Right.
Because that's what the skeptics always say, it's just a vitamin.
No, and that's a common misconception that really clouds the science. When we talk about vitamin C in this context, we aren't talking about nutritional replacement.
Okay.
We aren't trying to fix a dietary deficiency. We are talking about pharmaconutrition, or really using it as a drug. The doses we are talking about are pharmacological, not nutritional. The target population here is very specific.
And who is that?
Patients with sepsis who either have ARDS, acute respiratory distress syndrome, or are at very high risk for developing it.
And the stakes couldn't be higher. Sepsis is still a leading cause of mortality. And ARDS, well, if you've treated ARDS, you know the drill. We have great supportive care.
We do.
We have sophisticated ventilators. We have prone positioning. We have neuromuscular blockade. But pharmacologically?
Pharmacologically, we have almost nothing that treats the underlying injury. We are supportive, not curative. We are desperate for a drug that actually preserves the organ function at the cellular level.
And in the ASTER trial, the authors weren't just throwing vitamin C at inflammation broadly, right?
No, not at all. They had a very specific biological target in mind.
This is a key point. They weren't just hoping it would generally boost the immune system. They were hunting a specific villain.
Correct. They were looking at cell-free hemoglobin.
Okay, let's deep dive on that. Cell-free hemoglobin. So usually we want hemoglobin inside our red blood cells carrying oxygen. That's its job.
That's where it's supposed to be, yeah.
What happens when it gets out?
It becomes a toxin.
Yeah.
In sepsis, you have hemolysis, the breakdown of red blood cells.
Mm-hmm.
The systemic inflammation and the sheer stress from just, turbulent blood flow cause the red blood cells to pop.
There's rupture.
They rupture. That releases hemoglobin directly into the plasma. We call that cell-free hemoglobin. And it turns out cell-free hemoglobin is incredibly toxic to the vascular endothelium, the lining of your blood vessels.
So it's basically like acid in the pipes.
In a way, yes. Structurally, when hemoglobin is outside the protective environment of the red blood cell, it causes profound oxidative damage. It's an iron-containing molecule that just runs wild.
And what does that do?
Well, a couple of things. First, it has a high affinity for nitric oxide, so it scavenges the nitric oxide from your blood vessels.
And we need nitric oxide to keep vessels relaxed and open, so if it's gone...
You get vasoconstriction.
Mm-hmm.
But crucially for ARDS, this oxidative stress destroys the tight junctions between the endothelial cells. It increases vascular permeability.
It makes the vessels leaky.
It makes them leaky, like a garden hose full of holes.
And leaky vessels in the lungs mean fluid moves from the blood into the air spaces. That's pulmonary edema. That's ARDS.
Precisely. That is the pathophysiology. So the hypothesis for the ASTER trial was that high-dose 5E ascorbate could act as a scavenger.
Like a molecular sponge.
Exactly. Vitamin C is a potent antioxidant. The idea was that if we flood the system with huge concentrations of ascorbate, it can neutralize this oxidative stress caused by the cell-free hemoglobin. It can protect that endothelial lining and stop the organs from failing.
And they had good reason to think this, right? This wasn't just a hunch written on a napkin.
No, the preclinical data was very strong. The source material highlights studies where high-dose ascorbate mitigated endothelial injury in cell cultures. It improved the integrity of the epithelial barrier.
So in a petri dish, it worked.
It did. And in animal models of sepsis, it actually facilitated the resolution of lung injury. So biologically, the arrow was pointing in the right direction. There was a very strong rationale for this.
So the hypothesis is high-dose C neutralizes the cell-free hemoglobin toxicity, protects the vessels, and saves the organs. That's the pitch.
That's the pitch.
Now let's look at how they tested it. This brings us to Section 2, Study Design and Architecture.
This trial was conducted by the PETAL Network. That stands for the Prevention and Early Treatment of Acute Lung Injury Clinical Trials Network.
The PETAL Network, these are the heavyweights.
Absolutely. These are the premier academic centers for critical care research funded by the NHLBI. If the PETAL Network runs a trial, you pay attention because the methodology is going to be rigorous.
And they use something called the ASTER platform. Can you explain what a platform trial is for our listeners who might not be research nerds?
Sure. Traditionally, you do one trial for one drug. You recruit 300 people, randomize them to drug A or placebo, and run the trial. Then, if you want to test drug B, you have to start all over again.
A whole new protocol, a whole new placebo group.
A whole new everything. A platform trial allows you to test multiple therapies simultaneously against a shared control group. It's much more efficient. So ASTER was actually designed to test two therapies, ascorbate, which we were discussing today,
Right.
and acetaminophen.
Right. And just to be clear for everyone listening, the acetaminophen results were published separately. We are ignoring Tylenol for today. We are focused strictly on the ascorbate arm versus the placebo arm.
Correct. But knowing it was a platform trial explains why the structure is so robust. It was a Phase 2b, multicenter, randomized, double-blind, placebo-controlled trial.
Gold standard.
That is the gold standard design. You can't get better than that.
So who did they put in the bed? Who is the ASTER patient?
They needed adults, 18 plus. They had to have confirmed or suspected infection, so sepsis plus orders for antibiotics. But they also needed severity criteria.
Okay.
This wasn't for the patient on the floor with a urinary tract infection who was feeling a little feverish.
They needed to be sick.
Yes. They had to be critically ill. They had to have hypotension requiring vasopressors or respiratory failure.
And what did they define as respiratory failure?
It's pretty broad, actually. Invasive ventilation, non-invasive ventilation like BiPAP or high-flow oxygen greater than 6 liters per minute.
So these are patients in the ICU or on their way to the ICU immediately.
Exactly. And they had to catch them early. The window for enrollment was within 36 hours of seeking treatment.
That's a fairly tight window, but typical for these early intervention trials, right? You want to treat the fire while it's starting, not when the house is already burned down.
Right. If the endothelium is already destroyed, if the organs have already failed, no amount of antioxidant is going to bring it back. You have to intervene during the injury phase.
Okay, so 6 septic patients early in their course. Now let's talk about who they kept out, the exclusion criteria. You see the standard stuff like cirrhosis, chronic alcohol use, end-stage renal disease on dialysis. But there is one exclusion here that is fascinating and totally unique to vitamin C trials, the glucose exclusion.
This is critical safety detail, and it highlights one of the major operational headaches of using high-dose vitamin C.
What was it?
They excluded any condition requiring hourly blood glucose monitoring, like diabetic ketoacidosis, DKA, or hyperosmolar hypoglycemic state, HHS.
Why? Why does vitamin C care if you have DKA?
It's not that the vitamin C hurts the DKA per se. It's not making the diabetes worse biologically. It's that the vitamin C breaks the machinery we use to monitor it.
How so?
High-dose ascorbate causes electrochemical interference with point-of-care glucometers, the fingerstick machines.
I want to flag this because we are going to come back to it in the safety section. But essentially, the vitamin C looks like sugar to the machine.
Exactly. The test strips use an electrochemical reaction, an enzyme called glucose oxidase, to detect glucose. Ascorbic acid is structurally similar enough that it gets oxidized at the electrode in a similar way.
So it creates a current.
It creates a current that the machine interprets as sugar, so it falsely elevates the glucose reading.
So the machine says your sugar is 300, but it might actually be 60.
Right, and you can imagine the cascade of errors. Imagine you are a nurse, you stick the patient's finger, the machine reads 350 milli-GDL, you look at your sliding scale, insulin protocol.
And the protocol says give 8 units of insulin.
You give the insulin.
But the patient's real blood sugar was actually 80.
And now you've pushed them into profound hypoglycemia, seizures, coma, death. It is a potentially lethal error caused by the technology failure.
Wow.
So the ASTER trial leaders said, we cannot have patients in this trial who require hourly monitoring because we cannot trust the bedside machines.
That is a terrifying operational hurdle, and it shows the level of thought that went into the safety of this trial. Okay, so we have the design. Now we have to talk about the elephant in the room.
The enrollment numbers.
The enrollment numbers. This is the termination context. And honestly, this is the most important part of the story for understanding the statistics. The trial was designed to enroll about 300 patients per group.
That was the target, yeah. That would have given them plenty of power to see a difference in their primary outcome.
But they stopped.
They stopped.
When?
June 15th, 2022.
And how many patients did they have when they stopped?
79.
79 total.
Total. 40 in the ascorbate group, 39 in the placebo group.
That is small. That is a fraction of the intended sample. That's barely a pilot study in terms of numbers. Why did they pull the plug? Did they kill someone in the ASTER trial?
No, and that's the important distinction. The Data and Safety Monitoring Board, the DSMB, stopped ASTER based on data from another trial.
Another trial?
Yes. The Lovett trial lessening organ dysfunction with vitamin C was published around that time.
And Lovett was the bad news bear.
Lovett was the definitive harm signal. In patients with septic shock, the vitamin C group in Lovett had higher mortality and more persistent organ dysfunction compared to placebo. It was a really negative trial.
So the DSMB is sitting there looking at Lovett, which had hundreds and hundreds of patients showing harm, and they look at ASTER, which is testing a similar drug in a similar population.
And they made the ethical call. They said, we cannot ethically continue to enroll patients when a large, high-quality trial just showed this intervention might kill them. You can't expose people to that risk.
ASTER is collateral damage of Lovett.
In a way, yes. But it means we are left looking at a data set of 79 patients instead of 600. That changes everything about how we interpret the statistics.
Oh, so?
We are severely underpowered. A difference of 2 or 3 patients dying or living swings the percentages wildly. We have to be so careful about overinterpreting any finding.
Okay, keep that underpowered sticky note on your mental whiteboard. We are looking at a fragment of a trial.
A fragment, exactly.
Let's move to section 3, the intervention. What exactly were they giving these 40 people?
They went big. This is the high-dose concept.
Yeah.
This isn't the 1.5 grams you see in some of the other protocols.
Right, the HAT protocol.
Exactly. This wasn't that. They administered 50 milligram of intravenous ascorbate every 6 hours.
Okay, let's do some math for the folks at home. For a 100 kilogram patient, which is about 220 pounds, That's 5,000 milligrams.
5 grams.
5 grams every 6 hours.
That's 20 grams of vitamin C a day. Correct. They did cap the single dose at 9 grams, just for safety in very large patients. But yes, this is massive. For context, the nutritional requirement is like 75 to 90 milligrams a day.
So this is hundreds of times that.
Hundreds of times. This is a drug.
How long did they give it?
For 120 hours, that's 5 days.
That's longer than the citrusali trial, right? I recall that being 96 hours.
It is. Citrusali was 4 days or 96 hours. ASTER went for 5 days or until ICU discharge. They wanted to ensure they covered the critical window of organ failure. They wanted to saturate the system during the entire acute phase.
And to be clear, this was not the ATU therapy. We aren't mixing in hydrocortisone and thiamine here.
No, that's a crucial distinction. The HAT therapy trials often use lower doses of vitamin C, like 1.5 grams every 6 hours, combined with steroids and thiamine. ASTER is pure high-octane ascorbate monotherapy at 50 millijoule killer.
Got it. And the placebo.
A matching placebo. Volume matched 5% dextrose and water, D5W.
Okay. Before we get to the results, I want to circle back to that safety protocol regarding the glucose. Because if I'm a clinician listening to this and I decide to give high-dose C, I need to know how to not accidentally kill my patient with insulin.
Right.
How did ASTER handle this logistically?
They were extremely rigorous. Because of that interference issue we mentioned, point-of-care testing finger sticks were essentially prohibited for anyone in the trial.
Prohibited? So as a nurse, you can't check a sugar at the bedside?
No,
Go.
not allowed. Sites were instructed to use central laboratory monitoring for glucose.
The big machines in the basement lab.
Exactly. The central lab analyzers usually use a hexokinase method or other chemical methods that do not have this interference. They're immune to the ascorbate effect.
So if you are giving this drug, you cannot trust the bedside glucometer. You have to draw blood, put it in a tube, send it to the lab, and wait for the result.
Correct. Every single time. And they kept that restriction in place for 24 hours after the final dose, just to be sure the ascorbate had cleared from the blood.
That is a heavy logistical lift for nursing. That's a lot of sticks, a lot of tubes, a lot of waiting.
It is. It's a huge workflow change. But it worked. We'll see in the safety outcomes that there was no hypoglycemia. It proves that you can do it safely, but you have to have the resources and the discipline to ignore the finger stick.
Okay. We have the patients, we have the drug, we have the safety rails up. Let's open the envelope. Section 4. Primary outcomes. What happened?
First, let's check the baseline. Were the groups equal?
They were mostly well-balanced, but the paper notes the ascorbate group was maybe a little sicker.
Numerically, yes. Just a little. The ascorbate group had more patients on assisted ventilation at baseline, 43%, versus 31% in placebo. And slightly more of them had ARDS at baseline, 25%, 40s as 18%.
So the vitamin C group started a few yards further back from the finish line.
It wasn't statistically significant, but in a small trial of 79 people, those few extra sick patients can skew things. It's a bit of a headwind for the treatment group. You had to keep that in mind.
Fair. So the primary endpoint was organ support-free days to day 28. Basically, how many days were you alive and not attached to a machine, a ventilator, vasopressors, or dialysis?
Right. A really good patient-centered outcome.
So what was the number?
It was a wash.
A wash.
Completely neutral. The ascorbate group had a median of 20.5 organ support-free days. The placebo group had 19.0 days.
So a difference of 1.5 days.
With a p-value of 0.528.
Not even close to significance.
Not in the same zip code as statistical significance.
Okay. So in this small sample, stopped early, high-dose vitamin C did not magically liberate people from machines.
Correct. If you look at the components, ventilator-free days, vasopressor-free days, renal replacement-free days, there was no significant difference in any of them either.
So headline number 1, the trial failed its primary endpoint.
Yes. Based on the primary outcome, you would say this is a negative trial, just like the others. You could close the book right there.
But this is the deep dive. We don't stop at the abstract. Because when you turn the page to the secondary outcomes, things get weird.
Weird is the right word. Or maybe confusingly optimistic.
Yeah.
This is where the forensic accounting really matters.
Let's talk about Section 5, mechanisms, biomarkers, and secondary outcomes. Let's start with the one that matters most to the patient. Mortality.
This is the wait, what?
Yeah.
Moment of the paper. They looked at 90-day all-cause mortality. In the placebo group, mortality was 33%. 1 in 3 patients died.
Which is about what you'd expect for this population of septic patients with organ failure. That's not surprising.
No, that's a very believable number.
Okay. So what about the ascorbate group?
It was 15%.
Whoa. Wait, say that again?
33% of the placebo group, 15% in the ascorbate group.
That's a massive reduction. That's cutting the death rate by more than half.
It is a huge effect size.
Yeah.
A relative risk reduction of more than 50%. It's the kind of number that makes you sit up straight.
And the p-value. Please tell me the p-value was significant.
0.057.
Oh.
It just missed.
It missed by a whisker. 0.05 is the magic cutoff for statistical significance, and this was 0.057. So strictly speaking, scientifically, it is not statistically significant. At that trend, I mean, come on.
That trend is undeniable. The survival curves, the Kaplan-Meier curves, they separate early and they stay separated.
This is the opposite of Lovett. Lovett said vitamin C increased mortality. ASTER is saying, hey, maybe it decreases mortality.
Exactly. And this is the central tension of this paper. We have a trial stopped because of Lovett, showing a signal that completely contradicts Lovett.
But we have to be careful, right? N equals 79.
Extremely careful. With only 40 people in the treatment arm, a difference of just 2 or 3 deaths changes the percentage wildly. This could be random noise. This could be a fluke.
So we can't say vitamin C saves lives based on this?
Absolutely not. We cannot say that, but we also can't say it hurts them. The signal is pointing resolutely away from harm.
Okay, so clinical mortality looks arguably better. What about the biology? Did the drug do what they thought it would do? Did it lower the inflammation?
This is where it gets interesting, because this helps us adjudicate that mortality signal. If the mortality benefit is real, there should be a biological footprint.
It should show up in the blood work.
It should. They measured STNF-R1, soluble tumor necrosis factor receptor 1.
Which is a marker for what?
It's a marker of inflammation and TNF signaling activation. It's a very reliable barometer for how angry the immune system is.
Yeah.
High levels are bad.
Yeah.
It was significantly reduced in the ascorbate group.
Statistically significant.
Yes. P equals 0.005 at day 2, and P equals 0.008 at day 3. That is a very robust statistical finding.
So the vitamin C definitely did something. It dampened the inflammatory signal compared to placebo?
Without a doubt, it had a biological effect.
Okay, so we have a survival trend and a confirmed anti-inflammatory effect. But what about the specific target, the cell-free hemoglobin, the acid in the pipes that we spent 10 minutes talking about at the beginning?
This is where the hypothesis takes a hit.
Oh.
They measured cell-free hemoglobin levels. No difference.
Nothing.
Nothing significant. The levels didn't change significantly between groups.
So the drug worked on inflammation, potentially worked on survival, but not by the mechanism they designed the trial to test.
Precisely. It didn't seem to scavenge the hemoglobin, or at least it didn't lower the measurable levels.
Yeah.
But it did lower STNF-R1. So it's doing something, just maybe not the specific thing they enriched for.
That is classic medicine. We fix the patient, but we have no idea why.
Well, there might be a clue in another secondary outcome. This one is my favorite part of the paper because it connects to history.
Which one?
The diuretic effect.
They peed more?
A lot more.
How much is a lot?
By day 7, the cumulative urine output in the ascorbate group was about 2,588 milliliter. In the placebo group, 11,199 millivolt.
That is double. More than double. That's a massive difference.
And the total fluid output followed the same trend. The p-value was 0.0024 for urine and 0.006 for total fluid. It's a real finding.
Why does vitamin C make you pee? I've never heard of that.
It's actually an osmotic diuretic, and this has been known since the 1940s. The paper cites literature from 1940s suggesting ascorbate acts as a diuretic.
Get out of here.
It's true. When you give huge loads of this solute, remember, 20 grams a day, the kidneys have to filter it and flush it out, and water follows the solute. It's the same principle as giving mannitol for brain swelling.
So let's connect this to the bigger picture. If you have a patient with ARDS, wet lungs, and you have a drug that acts as a diuretic.
Exactly. We know from the famous FACT-TT trial that a dry lung is a happy lung, keeping ARDS patients on the dry side, or even improves oxygenation in ventilator-free days.
So is it possible that the benefit of vitamin C isn't some magic molecular antioxidant scavenging, but simply that it's a really expensive Lasix?
It is a very plausible theory. The authors discuss this explicitly. The diuretic effect might be the actual mechanism of action for any pulmonary benefit we are seeing. It's volume management. It's just making them less fluid overloaded.
That is hilarious and profound. We are looking for molecular magic, and it might just be peeing.
Sometimes the simplest physiological answer is the right one.
Let's wrap up the data with safety. We mentioned the glucose thing. Did anyone actually get hurt?
Despite the Levitt trial scare, ASTER showed no safety signals.
No hypoglycemia.
None reported. The central lab monitoring protocol worked perfectly.
What about kidney stones? Everyone always asks about oxalate stones with vitamin C. That's the classic concern.
Right. One patient in the ascorbate group developed the stone. 0 in the placebo group.
One patient out of 40.
One patient.
Yeah.
Not statistically significant. And no signals of oxalate nephropathy. That's kidney damage from crystals. So no acute kidney injury from it.
So in this small group, it appeared safe.
Yes. Safe, provided you control the glucose monitoring. That is the big asterisk.
Okay, so let's summarize part 1 before we let our listeners go. We have the ASTER ascorbate trial. N equals 79. Tiny. Stopped early because another trial, LOVEAT, showed harm.
Right.
Primary outcome. Organ support-free days. It was neutral, a wash, no difference.
Correct.
But UT, the secondary outcomes. 90-day mortality trended better cut in half, nearly significant at p=0.057, and inflammation, measured by STNFR1, went down significantly.
And the patients had significant diuresis, they had higher urine output, they peed more.
And the mechanism they thought was the key cell-free hemoglobin didn't move?
Correct. The original hypothesis didn't pan out.
And finally, it contradicts the very trial that shut it down.
That is the ultimate irony. Lovett says harm, ASTER says potential benefit.
It is a mess, a fascinating data-rich mess.
It creates a real conflict. We have citrus alley, which showed a mortality signal. We have Lovett, which showed a mortality harm. And now ASTER, which shows a mortality signal again.
Which leaves the clinician standing at the bedside asking, what on earth do I do?
And that is exactly what we're going to try to answer in part 2.
Coming up in the next deep dive, we are going to step back from the p-values and look at the patient. How do we reconcile these conflicting trials? Is there a Goldilocks patient who actually benefits?
We'll dig into whether the diuretic effect explains everything. We'll ask if there is a specific subphenotype, a certain kind of septic patient that we should be targeting.
And we will tackle the ultimate question. Should we ever prescribe high-dose vitamin C again, or should we close this chapter forever?
It's a puzzle, and we're going to try to solve it.
Join us for part 2. You won't want to miss it. Welcome back to the deep dive. If you were with us for part 1, you know we left things on a bit of a cliffhanger. We started unpacking the ASTER trial, this massive high-stakes investigation into whether high-dose vitamin C or ascorbate could actually save organs in patients with sepsis.
It's good to be back. And frankly, part 1 was just the setup. That was us laying out the tablecloth and polishing the silverware. Today we were serving the main course, and I have to warn you, it is a complicated, messy, and fascinating dish.
It really is. Just to quickly orient anyone jumping in now, though I highly recommend going back to part 1, we established that the primary goal, the mission of the study, was to see if this antioxidant cocktail could improve organ failure-free days. And the headline result regarding that specific mission was neutral.
It was a wash. Statistically speaking, for the primary outcome, the drug just didn't move the needle.
But, and this is a massive but, the story didn't end there. In fact, the context around how this trial ended is almost more dramatic than the data itself. We teased this at the end of the last deep dive. This trial didn't finish naturally.
No, it didn't.
It was halted. It was stopped in its tracks.
It was. And that is where we have to start today. Because usually, when a clinical trial is stopped early, it's for one of two reasons. Either the drug is working so incredibly well that it's unethical to withhold it from the placebo group.
Which is the dream scenario. That's the ticker tape parade moment.
Right. Or it's causing harm, and it's unethical to keep giving it.
And in the case of ASTER, it was the latter. That's what happened.
Well, it's nuanced. That's the key word for today.
Yeah.
Nuance. ASTER itself didn't find a smoking gun of harm, not initially, not within its own walls, but a completely different trial called Lovett Lessening Organ Dysfunction with Vitamin C. It came out while ASTER was still recruiting patients.
Oh, okay.
And Lovett dropped a bombshell.
This is the neighbor's house is on fire scenario.
Precisely. Lovett was a much larger trial, and their data suggested that patients getting the vitamin C cocktail were actually dying at a higher rate, and they had more persistent organ failure than those on placebo.
Wow. That is the nightmare. You're running your study, everything seems fine, and then this report lands on your desk from another group saying, hey, we think this stuff might be poisonous.
So the Data and Safety Monitoring Board, the DSMB for ASTER, had to make a tough call, a really tough call. They looked at the Lovett data, they looked at the slow enrollment in their own trial, and they essentially said, we cannot ethically continue.
So they pulled the plug.
They did. So ASTER, which was intended to enroll hundreds of people, over 300 per group, the plan was severed. They stopped with just 40 patients in the vitamin C group and 39 in the placebo group, a total of 79.
Which leaves us with a bit of a forensic crime scene, doesn't it? We have this truncated trial, a neutral primary outcome, but a huge cloud of suspicion from another study.
Hey.
So our mission for this Part 2 deep dive is to be the forensic accountants. We aren't just looking at, did it work? We need to act as investigators. We need to look at why it was stopped, whether it's actually dangerous, who it might help, and most importantly, the question I know everyone listening wants answered, what do we do on Monday morning?
That is the ultimate question. If you are an intensivist standing at the bedside tomorrow, is vitamin C in the trash bin, or is there still a flicker of hope?
So let's unpack this. I want to start with the safety aspect, because if Levitt said this stuff kills people, we need to know why. And looking through the ASTER source material, there is this bizarre, almost sci-fi side effect that kept popping up, the glucometer glitch.
It sounds like a plot point from a techno thriller, doesn't it? The glucometer glitch. But this is a very real, very dangerous chemical phenomena that I think a lot of people outside of clinical chemistry don't fully appreciate.
Break it down for us. How does a vitamin mess up a machine? I mean, a glucometer is supposed to measure sugar. Vitamin C is, it's a vitamin.
To understand the glitch, you have to understand the machine. In modern hospitals, we use point-of-care glucometers. These are the little handheld devices nurses use to check blood sugar right there at the bedside.
The finger prick ones.
The finger prick ones. They are fast, they are convenient, and they rely on electrochemistry.
Electrochemistry. Okay, take us to chemistry class.
Okay, so basically there's an enzyme on the test strip. When you put a drop of blood on it, that enzyme reacts with the glucose in the blood. That reaction, it releases electrons. It creates a tiny electrical current.
Right.
The machine measures the strength of that current and then translates it into a blood sugar number for the screen.
Okay, simple enough. More sugar, more reaction, more current, higher number.
That's the logic.
Yeah.
Until you introduce high-dose ascorbate. Ascorbic acid is an antioxidant. By definition, an antioxidant is a molecule that just loves to donate electrons. It oxidizes very, very easily. So when you have massive amounts of it in the blood, and remember, we are talking about pharmacological megadoses here, not an orange juice worth the ast. The ascorbate itself oxidizes on the test strip.
Oh, I see where this is going. The ascorbate is creating its own current.
It creates a massive current. The machine doesn't have a camera. It can't see that it's vitamin C. It just feels the electricity. So it assumes, wow, that's a lot of glucose.
And it gives you a reading of, say, 300 milli-GDL.
Or 400. Or even higher. It tells the doctor the patient has skyrocketing blood sugar.
But the patient doesn't actually have high blood sugar. It's fictitious hyperglycemia.
That's the clinical term for it. The real sugar is normal. Maybe it's 100. But the machine is screaming 300. Now put yourself in the shoes of a diligent ICU doctor or nurse. You see a blood sugar of 300 in a septic patient. What do?
You treat it. You follow the protocol. You order insulin.
You order insulin. And that is the trap. You give insulin to a patient whose blood sugar is actually normal. You drive their real glucose down from 100 to 40 or 30.
Oh, wow.
You induce severe hypoglycemia, which is deadly. In a septic patient, absolutely. It can cause seizures, arrhythmias, coma, and death. It removes the fuel from the brain. It's incredibly dangerous.
That is terrifying. It's an iatrogenic injury harm caused by the treatment itself, but mediated through a confused machine. It's like the GPS telling you to drive off a cliff.
That's a perfect analogy. And this is why the ASTER trial protocol was so fascinating to review. The authors of the ASTER paper were very, very clear on this. They knew about this risk. They explicitly prohibited the use of point-of-care glucometers for anyone in the study.
So they went old school. How did they check it?
They went central lab only. That means every time they needed to check a sugar, they had to draw a tube of blood, label it, tube it down to the main hospital laboratory, and run it on the big chemistry analyzers.
Why are the big machines better? Are they just smarter?
The large analyzers typically use a different method, often hexokinase-based spectrophotometry. They look at light absorption, not just an electrical current on a strip. They're much more specific. They aren't fooled by the vitamin C.
But think about the logistical hurdle there. I mean, if I'm a nurse, I'm used to getting a sugar check in 10 seconds. You're telling me now I have to wait 45 minutes to an hour for every single check.
It is a massive operational burden. It slows everything down. It requires incredible discipline from the entire team. But look at the result. In the ASTER trial, because they enforced this strict protocol, do you know how many hypoglycemic events they had?
I'm looking at the safety table here in the paper. It says 0.
0. Not 1. Now, compare that to the Levitt trial, the one that showed harm. In Levitt, While they warned about glucometers, the protocol might not have been as draconian, or perhaps in a larger real-world setting, compliance just slipped.
It's harder to control in a bigger study.
Much harder. And Lovett had a significantly higher rate of hypoglycemia in the treatment group.
So connecting the dots here, is it possible that the harm seen in other trials wasn't the vitamin C directly poisoning the organs, but rather this glucometer interference leading to accidental insulin overdoses?
It is a leading theory. Many experts speculate that the mortality signal in Lovett was actually a safety signal regarding glucose management. It implies the drug might not be biologically toxic, but it is logistically toxic.
Logistically toxic. That is a brilliant way to put it. If you cannot guarantee that every nurse and doctor on every shift knows not to use the handheld monitor, you are introducing a lethal risk.
Exactly. And in a strained healthcare system, relying on perfect adherence to a do not use the easy machine rule is a high-risk bet, a very high-risk bet.
Okay, so that covers the sugar issue, which is huge. But what about the kidneys? I feel like every time I hear about vitamin C, someone mentions kidney stones. It's like the urban legend of vitamins.
It's not entirely an urban legend. There is real chemistry there. Vitamin C is metabolized into oxalate. Oxalate is what makes up the most common type of kidney stones, calcium oxalate stones.
Ah, right.
So theoretically, if you pump someone full of vitamin C, you are loading their system with oxalate. The risk is real.
So did ASTER see an epidemic of kidney stones in their patients?
They looked for it diligently. They followed these patients out to 90 days, and they found exactly one patient in the ascorbate group who developed a stone.
1 out of 40.
And 0 in the placebo group. Statistically, that is meaningless. It's a 2.5% rate versus 0%. You can't draw a conclusion from an N of 1, but it certainly wasn't an explosion of stones.
And what about the liver? Any signs that the liver was struggling to process this? Any drug-induced liver injury?
No, none observed. They monitored liver enzymes, AST, ALT, bilirubin. There was no signal there whatsoever.
So if we wrap up the safety section, in the ASTER protocol, specifically with the central lab glucose monitoring, they did not see the harm signal that shut down the other trial.
Correct. Within the walls of this specific, highly controlled protocol, the drug appeared safe. But safe does not mean effective. You can drink a gallon of water safely, but it won't cure your sepsis. And that leads us to the next big question. Were they giving it to the right people?
Right. This brings us to the concept of predictive enrichment. I love this term. It sounds like something a financial advisor would sell me, but in medicine, it means finding the specific patients who are biologically primed to benefit from a drug.
We've moved past the era of one-size-fits-all for sepsis. We tried that for decades, and it has failed. Sepsis is too messy. It's too heterogeneous. It's like saying, I have a car problem. That could mean a flat tire, or it could mean the engine exploded. You don't use the same tool for both.
So the ASTER team had a hypothesis about which car problem vitamin C fixes.
They did. Their target was cell-free hemoglobin.
Let's unpack that. Hemoglobin is usually inside the red blood cell, right? That's its job.
Correct. Inside the cell, hemoglobin is the hero. It carries oxygen. It keeps us alive. But in sepsis, due to infection and inflammation, red blood cells can burst. This is called hemolysis. When they burst, they spill that hemoglobin out into the plasma.
And outside the cell, hemoglobin goes rogue.
It becomes a villain. Free hemoglobin is toxic. It scavenges nitric oxide, which you need to keep your blood vessels relaxed. It causes massive oxidative stress. It directly damages the endothelium, the inner lining of your blood vessels.
I've heard it described like rust. It effectively rusts the pipes of the vascular system.
That's a fair analogy. It causes oxidative corrosion. And the theory was that high-dose vitamin C, being a potent antioxidant, acts as a scavenger. It neutralizes this oxidative damage caused by the cell-free hemoglobin.
So the logic follows. If you have high levels of this toxic cell-free hemoglobin, you should benefit more from vitamin C. If you don't have it, the vitamin C has nothing to do. It's like bringing a fire extinguisher. It's only useful if there's a fire.
That was the hypothesis. So they measured baseline cell-free hemoglobin in all these patients before they gave the drug. They found that about 60% of them had high levels defined as greater than 10 milli-GDL.
That's a pretty big chunk of the population.
It is. So they compared the outcomes. Did the high hemoglobin group, the people with the fire, have a better response to the extinguisher?
I'm guessing, based on the tone of this deep dive, the answer is no.
The answer is, unfortunately, no. There was no interaction. The patients with high toxic hemoglobin levels didn't do any better with the treatment than the ones with low levels. The biomarker-guided approach, in this specific instance, failed to identify a responder group.
That is frustrating. It's such a clean biological theory. Hemoglobin causes rust. Vitamin C is the rust remover. Why didn't it translate?
That's the million-dollar question in translational medicine. Just because the mechanism works in a test tube or a mouse doesn't mean it drives the clinical outcome in a complex human with sepsis. Maybe cell-free hemoglobin is bad, but maybe it's not the main thing killing the patient.
Or maybe the dose wasn't high enough, or the timing was wrong.
All possible. Or maybe by the time you give the vitamin C, the damage to the endothelium is already done. You can't unrust the pipe once it's burst.
But wait, you mentioned earlier that the drug did do something biologically. It wasn't completely inert, right? It wasn't just expensive water.
No, it definitely had biological activity. And we know this because they measured another biomarker called STN-FR1.
Soluble tumor necrosis factor receptor 1. I'm definitely not saying that again. Let's stick to the acronym.
Good call. STN-FR1 is essentially a marker of inflammation. Specifically, it reflects the activation of the TNF pathway, which is a major driver to the cytokine storm in sepsis. Think of it as a thermometer for the immune system's anger.
Okay, so it gauges how hot the fire is burning.
And in the ascorbate group, levels of STN-FR1 dropped significantly at day 2 and day 3 compared to placebo.
Significant as in statistical significance, like a real...
Oh, highly significant. P-values of 0.005 and 0.008. The drug was biologically active. It dampened inflammation. It cooled the fire.
But, and I feel like I keep saying but, that drop in inflammation didn't turn into saved organs?
It did not. And this is a recurring, almost heartbreaking theme in critical care research.
Yeah.
We call it dissociation. We can fix the numbers, we can lower the inflammation markers, we can fix the physiology, but the patient still stays on the ventilator for the same amount of time.
It implies that the pathway we are modifying might not be the rate-limiting step for recovery. It's like fixing the air conditioning in a car that has a blown transmission. The car is cooler, but it still won't drive.
That is a perfect analogy. We cooled down the inflammation, but the engine, the organ failure, didn't restart any faster.
Now, speaking of fixing physiology, there was one finding in this study that really caught my eye. It wasn't the primary outcome, and it wasn't the inflammation. It was the fluid, the urine output.
Ah, yes, the diuretic effect. This is where things get really interesting. And frankly, it opens up a whole new can of worms regarding what we think this drug is doing versus what it is actually doing.
Walk us through the numbers here, because looking at the on-study variables table in the paper, there's a massive difference in how much fluid these patients were putting out.
Right. So in sepsis trials, we always track fluid in and fluid out. You pump fluids in to keep blood pressure up, but then you want the kidneys to pee it out so the lungs don't get soggy.
Right, fluid balance.
In ASTER, the total fluid intake was similar between the two groups. They both got about the same amount of saline and meds.
But the output?
The output was drastically different. By day 7, the placebo group had put out about 1.2 liters of urine and fluid. The vitamin C group, about 2.5 liters.
That is more than double. That's not a rounding error. That's a real effect.
No, it's a significant physiological effect. The patients getting vitamin C were diuresing. They were peeing significantly more.
Is vitamin C a diuretic? I thought it was an antioxidant. I didn't know it acted like a water pill.
Well, interestingly, this isn't a new discovery. If you dig into the archives, there are reports from the 1940s suggesting that ascorbate causes diuresis. It creates an osmotic load, it's a solute that the kidneys have to clear, and it drags water with it.
Osmosis 101. Water follows salt, or in this case, water follows the vitamin.
Exactly. And this matters because we know that in conditions like ARDS, acute respiratory distress syndrome, keeping the lungs dry is generally good. The FACT-CT trial, a famous study from years ago, proved that a conservative fluid strategy gets people off ventilators faster.
So, hold on. If the vitamin C group had any trend toward benefit, and we'll get to the mortality numbers in a minute, could it just be because they had less fluid in their lungs?
That is a very valid hypothesis. It creates a massive confounding variable. Are we seeing the effect of a sophisticated molecular antioxidant scrubbing the endothelium? Or are we just seeing the effect of volume management?
Are we just giving them a really, really expensive version of Lasix?
It's possible. And if it's just volume management, we have much cheaper standard drugs to achieve that, like furosemide. And furosemide doesn't break your glucometer.
It really highlights the complexity of these trials, doesn't it? You think you're testing mechanism A, but the drug is secretly doing mechanism B in the background.
It's a classic case of mechanism bias. We fall in love with the complex story and miss the simple one. The drug made them pee. That might be the most important physiological change it caused in the whole study.
Fascinating. Okay, I want to pivot to the limitations. Because we've been talking about this data like it's gospel, but we have to remember this was a small study.
Small is putting it gently. This was a truncated study. a fragment of a study.
Right. They wanted 300 plus people. They got 79. What does that do to the math, to the statistics?
It destroys the power of the study. In statistics, power is your ability to detect a difference if one actually exists. With only 79 people, you can only detect massive, slap-you-in-the-face differences.
Okay.
If the drug helps a little bit, say it improves survival by 5%, study of this size will miss it completely. It'll look like noise.
So a neutral result here doesn't necessarily mean it doesn't work. It might just mean we didn't have enough people to prove it works.
Correct. Absence of evidence is not evidence of absence. However, on the flip side, small studies are also prone to what we call type 1 error false positives, or just random fluctuations that look like signals.
Because if you flip a coin 10 times, getting 7 heads is weird, but possible. If you flip it 1,000 times, getting 700 heads is almost impossible.
Exactly. With small numbers, luck plays a huge role. And that brings us to the baseline characteristics. When you have small groups, it's really hard to balance them perfectly through randomization.
And I noticed that looking at Table 1, the vitamin C group looked sicker.
They were numerically sicker. Look at the baseline ventilation status. In the ascorbate group, 43% were on assisted ventilation at the start. In the placebo group, only 31%.
That's a big difference.
It is. The ascorbate group also had more ARDS at baseline, 25%, ADS 18%.
So the vitamin C group started the race 10 yards behind the starting line.
They did. They were sicker. And yet, and this is where we get into the mortality paradox, fewer of them died.
This is the part that makes my head spin. Let's look at the numbers. Primary outcome. Neutral, organ failure-free days, neutral. But 90-day mortality.
90-day mortality was 15% in the vitamin C group versus 33% in the placebo group.
That is huge difference. That's less than half the death rate. If I see those numbers, I'm thinking, give me the drug.
It is numerically striking. I mean, it jumps off the page. The p-value was 0.057.
Which is just barely above the magic 0.05 cutoff for statistical significance. It's borderline. It's a trend.
It is a strong trend. But we have to be incredibly careful here. We cannot let our emotions or our hope override the statistical rigor.
Why not? If I'm a patient, I care about dying more than I care about organ failure-free days. If the drug cuts death in half, why are we not celebrating? Why isn't this the headline?
There's several reasons. First, mortality was a secondary outcome, not the primary. You can't change the goalposts after the game is played. That's a cardinal rule of trials. You pre-specify your main goal. Second, the sample size is tiny. A difference of just 2 or 3 deaths shifts those percentages wildly. If just 2 people in the vitamin C group had died instead of lived, the difference vanishes. It's fragile.
In third,
and this is the big one, it contradicts the larger, more robust trials. The Lovett trial, which had nearly 900 patients, showed higher mortality with vitamin C. The Citrus Alley trial, another medium-sized one, showed lower mortality, just like ASTER. So we have conflicting signals all over the place.
it's whiplash. One study says it saves you, one says it kills you, one says it does nothing.
And that inconsistency is usually a sign that the effect isn't real, or at least not robust. If a drug is a miracle cure like penicillin, every trial shows it works. The arrow always points up. When you see this flip-flopping, sometimes it helps, sometimes it harms, sometimes it's neutral, it usually suggests that the intervention isn't the main driver of the outcome.
Or that the conditions under which it works are so narrow that we just haven't figured them out yet.
Or that chance is playing a huge role. In the ASTER trial, maybe the placebo group just happened to have a run of bad luck. Maybe those patients had unmeasured comorbidities that made them more likely to die. With only 39 people, that happens.
Speaking of comorbidities, we need to talk about who was left out of this party, because clinical trials often pick the perfect patients, and ASTER was no exception.
ASTER was very picky, and for good safety reasons, I should add. But the exclusion list is long. No cirrhosis, no end-stage renal disease, no kidney transplants, no uncontrolled diabetes.
Wait, no uncontrolled diabetes? That's because of the glucometer issue, right?
Exactly. They couldn't risk it. But think about the average ICU population. How many patients have diabetes, kidney issues, or liver issues?
That sounds like half the ICU, maybe more.
It sounds like the majority of the ICU.
Mm-hmm.
The thickest patients, the ones with liver failure and kidney failure, are often the ones with sepsis. So even if ASTER had been a resounding success, we wouldn't know if it works for the liver failure patient in bed 4.
That is a huge limitation for generalizability. We have a drug that, at best, works for a specific slice of the population that doesn't have chronic organ failure.
Right. And honestly, the safety profile we discussed earlier relies on these exclusions. If you gave this high dose of oxalate-producing vitamin C to a patient with renal failure who can't clear it, you might see disaster. The study protected itself by excluding the people most likely to be harmed.
Which is good science for a phase 2 trial. You want a clean signal. But bad news for real-world application where patients are messy.
Exactly. Real-world patients have baggage. Lots of baggage.
So we're approaching the end of our forensic analysis here. Let's recap the evidence on our desk. We have a drug that cleared inflammation. We know STNFR1 went down.
Sure.
It made people pee. Diuresis went up.
Sure.
It had a glitch with glucometers that necessitated a strict, difficult protocol.
A big, dangerous check.
It failed its primary outcome. Organs didn't improve. But it showed this tantalizing, perhaps illusory survival signal in a small group.
That is a perfect summary of the chaos.
So now we have to land the plane, the Monday morning verdict. I want you to put yourself in the shoes of a listener who is an ICU physician. It's Monday morning. You have a patient admitted with severe pneumonia, sepsis, respiratory failure. They look like an ASTER patient. Do you order the high-dose vitamin C?
The short answer is no. Absolutely not.
Walk me through the rationale. Why no, despite that 15% mortality number that's so tempting?
It comes down to the hierarchy of evidence and the principle of do no harm. First, the primary endpoint failed. The study did not prove that the drug prevents organ failure. That is the legal fact of the trial. We cannot ignore it.
Okay, strike one.
Second, we have conflicting data from a larger trial that showed potential harm. When you have one small trial hinting at benefit and one large trial shouting danger, you have to respect the danger signal. You can't cherry-pick the data you like.
Strike two. You can't ignore the neighbor's house burning down just because your kitchen is okay for now.
Exactly. And third, the operational risk. To give this drug safely, based on ASTER, I have to institute a central lab-only glucose protocol. I have to tell my nurses, do not touch the glucometer.
And what happens at 3 a.m.?
At 3 a.m., when the unit is busy and a covering nurse who wasn't at the morning briefing walks in, systems fail. Habits kick in. Someone will check a finger stick. Someone will see a high number. Someone will give insulin. And if that happens, I risk killing my patient with hypoglycemia.
And doing that for a drug with unproven benefit
Is bad medicine.
Yeah.
The risk-benefit ratio is completely off. If this were a cure for Ebola with a 90% survival benefit, I'd take the glucometer risk. I'd stand by the bedside myself to guard the machine. But for a drug that failed its primary outcome, no way.
So it's not just that the data isn't good enough. It's that the cost of safety, the operational tax, so to speak, is too high for the potential lack of reward.
Precisely. It remains an experimental therapy. It is not standard of care.
Does that mean the door's closed forever? Is vitamin C dead in the ICU?
Science never really closes the door forever. But it is heavily boarded up right now. The harm signal in Alivati makes it ethically very difficult to start a new massive trial without a major redesign.
What would that redesign look like?
You would need foolproof continuous glucose monitoring that doesn't rely on the strip chemistry technology that is getting better but isn't everywhere yet. You would need to really target a specific subgroup, maybe based on that inflammation marker we discussed. But honestly, the funding agencies are likely to look at this conflicting mess and say, let's put our money elsewhere.
That's a sobering reality. We spent decades chasing this magic bullet, the vitamin C, thiamine, steroid cocktail, the Merrick protocol, all of it. And piece by piece, the rigorous trials have kind of dismantled it.
That is the scientific process. It's painful. It's disappointing. We all wanted it to work. I wanted it to work. A cheap, safe vitamin that saves lives. Who wouldn't want that? It would be a global health miracle. But we can't let our desire for a cure override the data. A failed trial isn't a failure if it stops us from using a treatment that doesn't work or, worse, hurts people.
That's a great perspective. ASTER didn't give us the answer we wanted, but it gave us an answer we needed.
And it taught us about glucometers. That alone might save lives, just knowing that interaction exists.
Absolutely. I want to leave the listeners with a provocative thought before we hit the summary. We talked about the diuretic effect.
Yes, the 2.5 liters of urine.
If the diuretic effect is real, have we been studying a complex antioxidant mechanism for decades, writing papers about free radicals and electron scavenging, when we should have just been looking at how vitamin C affects the kidneys?
It's a classic scientific blind spot, isn't it? We love the fancy molecular story. We want it to be about scavenging electrons. But sometimes the drug is just making you pee. And sometimes the mechanism you think you're testing isn't the one doing the work. If vitamin C ever makes a comeback, I bet it will be investigated as a modulator of fluid balance, not just an antioxidant.
A fascinating thought to chew on. All right, let's wrap this up. For the commuter pulling into the parking lot or the person finishing their workout, here are the 5 key takeaways from our deep dive into the ASTER Part 2 analysis.
Let's hear them.
Number 1, primary result. High-dose vitamin C did not improve days alive and free of organ support compared to placebo in septic patients. The main mission failed.
Clear and simple.
Number 2, safety and glucose. The trial had 0 hypoglycemia events, but only because they strictly prohibited point-of-care glucometers. These devices read falsely high in the presence of vitamin C, creating a dangerous trap for clinicians.
The critical safety pearl.
Number 3, mortality signal. Despite the neutral primary outcome, 90-day mortality was numerically lower in the vitamin C group 15% versus 33%. However, this was not statistically significant, relied on a small sample size, and flatly contradicts the larger ELVAPATE trial, which showed harm.
The paradox remains unsolved.
Number 4, biomarkers and physiology. Vitamin C wasn't inert. It significantly reduced STNFR1, an inflammation marker, and increased urine output, proving it has biological activity, even if clinical outcomes weren't met.
It did something, just maybe not the right thing.
And finally, number 5, clinical verdict. Due to conflicting data across trials, the risk of glucose measurement errors, and the termination of this study, high-dose vitamin C should not be adopted for routine use in sepsis on Monday morning.
Agreed. Keep it in the research journals, not the IV bags.
And with that, we are closing the book on the ASTER trial. It's been a wild ride through statistics, biochemistry, and broken glucometers.
It's never boring in the deep dive.
Never. Thanks for listening, everyone. Stay curious, check your glucose sources, and we'll see you on the next deep dive.
Goodbye, everyone.